second of the day, you are surrounded by millions of bacteria. They're on the things you touch, the water you drink, and the air you breathe. With so many bacteria surrounding you at every moment, you have a high chance of catching a disease. Diseases can be caused by 3 types of organisms: bacteria, viruses, and fungi. Luckily for you, your body has a built-in defense mechanism called your immune system to protect you from these diseases. There are 2 levels of immune responses: innate and adaptive. Innate immunity consists of nonspecific immune responses, meaning each macromolecule or cell involved can act on all kinds of pathogens. Pathogens are anything that causes a disease. Adaptive immunity is the immune responses by cells that are specific to a certain pathogen. The first line of nonspecific defense your body has is a barrier that helps prevent pathogens from entering the body. Barriers include skin, mucous membrane, cilia in your respiratory system, and stomach acid. If the first line of defense is breached, your body initiates the second line of defense, whose function is to limit the spread of pathogens before adaptive immunity is triggered. The second line has 5 parts: phagocytes, inflammatory response, complements, interferons, and natural killer cells. Phagocytes are cells that ingest invading fungal and bacterial microbes. There are 2 types of phagocytes: neutrophils and macrophages. They migrate to infected areas in response to chemical attractants called chemokines. This process is called chemotaxis. Neutrophils engulf microbes and die in a few days. On the other hand, macrophages last for more time and can engulf a greater amount of microbes. They extend pseudopods and engulf large amounts of microbes and digest them with the combination of lysozyme, superoxide anion, and nitric oxide. Another factor in the second line of defense is the inflammatory response. The inflammations or swellings we see when we are sick are caused by the enlargement of blood vessels, a process called vasodilation. Vasodilation is induced by the compound histamine to increase blood supply to the area of infection, delivering a rush of phagocytes to fight the invaders. Lipid compounds called prostaglandins further promote blood flow to the area. There are also a group of proteins, collectively called complements, that induce the lysis or bursting of invading cells. Interferons, a group of signal proteins, block cell-to-cell viral infections. Natural killer cells destroy virus-infected body cells by attacking their cell membranes, causing them to lyse and die. If the second line of defense is not enough to defeat the disease, a third, adaptive immunity, is activated. Adaptive immunity relies on B lymphocytes and T lymphocytes. There are 2 types of T cells: cytotoxic T cell, aka TC cells, and helper T cells, aka TH cells. B and T lymphocytes are created in stem cells in the bone marrow, and when they mature, they circulate in the blood, lymph, and lymphatic tissue. Lymphatic tissues are spleen, lymph nodes, tonsils, and adenoids. Each B and T cell displays specificity for one particular antigen or epitope, meaning they can only recognize, bind, and act on one specific type of antigen. Antigens are the proteins found on the surface of the pathogens. An epitope is a part of an antigen that is recognized by the immune system. There are 3 phases in adaptive immunity: the recognition, activation, and effector cells. In recognition, antigen receptors in B and T lymphocytes recognize specific antigens or their epitopes. During activation, the binding of antigen receptors activates B and T cells and causes rapid cell division. This forms large populations of effector cells and memory cells. In the effector phase, B cells produce humoral response, meaning they produce antibodies, while T cells engage in cell-mediated response. For a B or T cell to recognize an antigen, the antigen has to be presented to them by an antigen-presenting cell. First, the pathogen or part of a pathogen is taken up by a macrophage or a dendritic cell, and its enzymes cleave the antigen into smaller peptides called antigen fragments or epitopes. The antigen fragments bind to MHC2 or major histocompatibility complexes in the cell. The MHC and bound antigen fragment then move up to the infected cell's surface. At this point, the macrophage or dendritic cell is called an antigen-presenting cell, or APC. If the APC encounters a T cell with the right specificity, the antigen receptor on the T cell binds to the MHC plus antigen fragment. At the same time, an accessory protein called CD4 on the helper T cell surface binds to the class II MHC, which helps keep the two cells joined. The binding promotes the secretion of cytokines called interleukin-1 from the antigen-presenting cells. Interleukin-1 binds to the helper T cell surface receptors. This activates a secondary messenger inside the cell called tyrosine kinase, inducing a signal transduction pathway, stimulating the cell to produce its own cytokines called interleukin-2. These cytokines and the cytokines from the APC activate the helper T cell and stimulate its replication into multiple clone cells. Clones of activated helper T cells release cytokines that initiate a signal transduction pathway in B cells, cytotoxic C cells, and other helper T cells specific to the antigen which activates them. Activated T and B cells then proliferate into effector cells and memory cells. Effector cells initiate immune responses to kill the current invading pathogens, while memory cells are able to rapidly respond to any exposure of the same antigen many years later.
The immune response of activated cytotoxic T cells is to kill already infected body cells. When normal cells are infected, they produce foreign proteins directed by the ingested pathogen's DNA. These proteins bind to class I MHC molecules, and the entire complex is displayed on the infected cell's surface. Class I MHCs mark the cell as a normal cell, while class II MHCs mark specialized cells, such as dendritic cells, macrophages, B cells, and T cells. Activated cytotoxic T cells, specific to the presented antigen, binds to the MHC antigen fragment complex with the help of accessory proteins called CD8. This binding induces a signal transduction pathway in the T cell, which causes the T cell to secrete perforin molecules and granzymes. Perforin is a protein that forms pores in the infected cell's membrane. Granzymes, enzymes that break down proteins, enter the infected cells by endocytosis and initiate the fragmentation of the nucleus and cytoplasm. This eventually leads to apoptosis, aka cell death. An activated B cell goes through a different immune response. First, an antigen specific to the B cell binds to the receptors on the surface of the B cell, and the cell takes in a few antigen fragments by receptor-mediated endocytosis. The class II MHC protein of the B cell binds to the fragments and presents them on the B cell's surface. The clones of activated helper T cells that have antigen receptors specific to the displayed fragment bind to the B cell and release cytokines which activates the B cell. The activated B cell multiplies into thousands of plasma cells and memory B cells. The plasma cells become antibody-producing factories, producing and secreting approximately 2,000 antibodies every second. The secreted antibodies bind to antigens and interfere with pathogen activity on mark pathogens for inactivation or destruction. One method that is employed is neutralization. Some antibodies bind to proteins on surface of virus, which prevents infection of host cells. Others bind to toxins released in body fluids, preventing toxins from entering body cells. Another method is opsonization, in which antibodies bound to antigens serve as a marker to signal macrophages or neutrophils to induce phagocytosis, which we previously identified as an innate immune response. Because each antibody has two antigen binding sites, they can link pathogen cells into aggregates and facilitate large-scale phagocytosis. The more phagocytosis that happens, the more APCs exist, and thus the more B and T cells created. In this way, immune response is a cycle that depends on positive feedback to be effective. Antibodies can also kill invading cells by teaming up with the complement protein complex. The binding of complement proteins is an antigen-antibody complex on a pathogen cell induces the generation of a membrane attack complex. The membrane attack complex forms a pore in the membrane of the cell, letting ions and water enter the cell, causing it to swell and lyse. The last method that antibodies use to kill pathogens is by calling natural killer cells. When a normal body cell is infected, the virus uses the cell's organelles to produce viral proteins. Sometimes these viral proteins can appear on the surface of the cell. When an antibody binds to it, its presence can signal a natural killer cell. The natural killer cell releases proteins that cause the infected cell to undergo apoptosis. Not only is your immune system efficient, it is also proactive. Your immune system is ready to respond even swifter and stronger when your body fluids are reinfected by a pathogen encountered previously. This is called a secondary immune response. The second exposure of antigens does not need to be engulfed by an antigen-presenting cell and presented to the helper T cells to start an immune response. Instead, they directly stimulate the memory helper T cells, which in turn activate the memory B cells and memory cytotoxic T cells. This makes secondary immune response faster and more effective than the first time. The immunological memory and secondary immune response is why people don't get some diseases like chickenpox for a second time.